Hi, welcome back to BIPN 140. This is our final set of LOs, and it's all about the senses. So we're going to revisit a lot of what we learned previously about how neurons work and talk about how they affect our senses, which is how we interact with the world. So we're only going to be doing two senses, vision and olfaction, and that's because they are interesting in different ways. They make use of very different types of sensory input, and there are some differences, but also some really basic commonalities that are true across all senses. So let's go ahead and dive in with vision. How do we sense things? Basically, right, every what is sensing? It's taking stimuli from the outside world and they affect our body in a certain way and they cause a certain perception. So it all starts with a sensory receptor. Um, this could be a cell, this could be a receptor in a cell, like a GPCR or something. And these collect and what's called transduce physical signals into neural electrical signals. And this happens in the sensory organs. So in the case of a lollipop, that would be the tongue, right? So transduce means to change something from one form into another. So it's changing a physical signal into an electrical signal. And that creates an electrical response. And then further on in the brain, deeper in the brain, we process and interpret these signals to give them meaning. So in this case, it's the meaning of sweetness. And that meaning allows us to act appropriately on this signal, whether we want to eat it or not. So in this sense, we're going to be, in this cellular neurobiology course, we're going to be talking about the sensory transduction. So what happens in the first cell that encounters the stimulus to turn that physical stimulus into an electrical signal. In some other courses like systems neuroscience, it's more about how those signals are processed and interpreted, which can be very, very complicated. Okay. So talking about that sensory reception and transduction. So the sensory receptor's function is to turn that outside stimulus into a change in membrane potential and thus a change in neurotransmitter release. And when you do that, that tells the next cell and the next cell and the next cell what is going on in the world. This sensory receptor terminology, I want to do a note for it. It's different for different senses. So usually the word receptor, that's a word we've heard a lot. It means like the channel or the molecule that detects the stimulus. So like a GPCR, R is a receptor, or a AMPAR receptor, right? However, in certain cases, especially in this case of sensory cells, it means the whole cell or neuron that does that. So we'll be talking about photoreceptors today. We're not talking about the one molecule that detects light. We're talking about the entire cell that detects light and sends that signal on. Just be aware that the word receptor can have both of these meanings. Okay. So what's the big idea here when we're talking about phototransduction? Well, the big idea is we're talking about light that is coming into our eyes and turning that light signal into a change in neurotransmitter release. So we're going to turn that external stimulus, that photons in the light, into a change in membrane potential in the photoreceptor, and thus a change in the amount of neurotransmitter a neuron releases. After we do that, the next neuron, and actually the retina itself is incredibly complicated, not to mention the whole brain, like half your brain almost is devoted to processing sensory stimuli of some sort. But it all starts right in our retina, in our photoreceptors, those neurons getting the light information and changing their neurotransmitter release to tell the whole rest of the brain what is going on in the visual world. Okay, so this is your eyeball. This is your retina. The light comes in and it hits your retina. The retina has certain photoreceptor cells, cones and rods. that detect that light. So those are the cells that we're going to be talking about today. So the rods detect all light and they work especially well in low light conditions. The cones, there are three types of cones, red sensitive, green sensitive, and blue sensitive. And so those are sensitive to specific wavelengths or colors of light, and they tell you about information about colors. And these photoreceptors will um, send signals to all sorts of different other cells that will eventually send their axons to the brain and tell your brain what is going on. Okay, so as I said, we're going to be focusing on the photoreceptors and how they work. First, let's start off in the darkness because there's stuff going on in the photoreceptors, even when it's dark. So when it's dark, there are two different channels open in the photoreceptors. We have our K plus lead channels that are always open. Okay, we knew about those. We also have cation channels, and these let in sodium and they let in calcium, and they're gated by extracellular GMP. And in the dark, there is a lot of, sorry, intracellular GMP, and there is a lot of that around. So thinking about that, how will these channels, when they're open, like they both are in the dark, affect the photoreceptor's membrane potential? Go ahead, think about that, and make your choice. Okay, so knowing what you know from every other cell that we've talked about, right? We have it that the cation channels are going to let cations into the cell. That's an inward current that is going to tend to cause depolarization, while the potassium leak channels, that is an outward current that is going to tend towards hyperpolarization. And these oppose each other, right? So we know that in typical neurons that only have the potassium leak channels open at rest, the resting membrane potential is quite low, like negative 60-ish. But because in the dark, these photoreceptors have this cation channel open, the resting membrane potential of a photoreceptor is higher than that. It's relatively high, around negative 40 millivolts. You might think at this point, well, why doesn't it spike? Isn't that usually above threshold? Well, I'm going to tell you this. These neurons don't make action potentials. They're sort of weird that way. So we'll talk about what they do. But just for now, no, because of the contributions of these two channels, the resting membrane potential is relatively high. Okay, now what happens when light comes in, when the photons are coming in? What do? So the photoreceptors contain a molecule called retinal that is coupled to a protein called an opsin. In rods, that opsin is called rhodopsin. In cones, there are three types of opsins. Remember, there were three types of cones. Each one has its own type of opsin, and they're sensitive to red-green or red, blue-green or red wavelengths corresponding to the type of the cone. So what does this retinal do? So it's a molecule that looks like this. When the retinal absorbs light, that light affects specifically this bond here, and it kind of like makes it flip around. So it goes from cis to trans. If you've taken OCHEM, you know what it means. That means if you haven't, don't worry about it. The key thing here is that it changes shape from this kind of bent shape to this kind of straight shape. Okay, why does that matter? It matters because the retinal changing shape, the retinal is kind of embedded in the opsin. So when it changes shapes, it activates the opsin. And that's true of all the opsins and all the different types of cones and rods. The opsin then will go and activate a G protein called transducin. Okay, so what's up with transducin? Well, transducin is a G protein, but it's very different. Not very different. It's a little bit different from the other G proteins that we've talked about. It's not like a typical G protein. So let's see, what does it do? So this is the opsin with the retinal. This is the transducin. So transducin is a heterotrimeric G protein. It has the three subunits, an alpha, a beta, and a gamma. And at rest, it's bound to a GDP. And then when the opsin with the retinal comes and when the retinal is stimulated by light, that's what this star here is for, then just like a usual G protein. On the alpha subunit, the GDP is switched for GTP, and the alpha subunit comes off away from the beta and the gamma subunit. Okay. Here it is the alpha subunit that is the most important, does the most important stuff. So when the alpha subunit is activated, it goes and it activates a molecule called phosphodiesterase or PDE. Phosphodiesterase then hydrolyzes uh, cyclic GMP to just regular GMP. So it like unmakes it cyclic. And what in effect does that do? It lowers the numbers of cyclic GMP by turning it into just regular GMP. Okay, so when this is activated, it activates the PDE, which destroys cyclic GMP. Okay. And eventually, how do you end this? Eventually, just like with all G proteins, the GTP is hydrolyzed to GDP, and the transduction is back in its inactivated state, and you stop that signaling. Okay. But for now, just know that when you have light, the opsin with the retinal activates transducin, which activates PDE, which lowers the levels of cyclic GMP. Okay, so putting it together, how does that affect phototransduction? So initially it's dark, there's cGMP. That cGMP opens that cation channel. In light, that retinal changes shape, and that activates this opsin. In this case, this is a rod, so it's rhodopsin. Then transducin switches the GDP for GTP. The transducin is activated. When it's activated, that alpha subunit of transducin will activate the PDE, the phosphodiesterase, and the phosphodiesterase will hydrolyze cyclic GMP and just turn it into regular GMP. With the cyclic GMP no longer around, the channel will close. So when there is light, and here I'm showing you what happens in the dark, but when there is light, what will happen to the membrane potential of the photoreceptors? All right, actually, it will hyperpolarize, right? Because this cation channel will close, but there's still the leak channels, the potassium leak channels. So these channels close and it's hyperpolarized. And so this shows the membrane potential in response to a light flash. And if it's just a little bit of light, a very weak light, then you get a very light hyperpolarization. If you have a very strong flash of light, it hyperpolarizes quite a bit. And that's what these traces are showing. They're showing the response to different kind of intensity of light flashes. But overall, the photoreceptors will respond to light. You're like, okay, well, why? Well, sort of, this is just how it is. You can also think of it in various ways, but actually before we talk about that, let's talk about this. Are these responses to light action potentials? No, you've seen action potentials. Action potentials don't look like that. Um, and as I mentioned before, photoreceptors are these weird neurons that don't make action potentials. These are purely hyperpolarizing responses. Okay. I will say what they do is release neurotransmitter when they are depolarized, similar to other neurons. So remember at the synaptic terminal, right, we have voltage-gated calcium channels, and at depolarized voltages, those channels are open and trigger the release of vesicles. That is still true in these neurons. So they release neurotransmitter when they are depolarized. So when they're hyperpolarized, it means they make less neurotransmitter release. So let me ask you something. How would this stimulus, literally a black dot, change the neurotransmitter release of a photoreceptor that saw it? What do you think? It would increase it. Okay, it's a dark spot. So don't think of like the light level of an environment being all or nothing. Think about like you're reading a page, okay? And the page has dark spots and the page has light spots, right? Which is like the paper or the screen. And so some of your photoreceptors are being exposed to dark spots, and some of them are being exposed to white spots, which is, the screen. So this dark spot, photoreceptors release neurotransmitters in the dark, meaning any of your photoreceptors that are currently being exposed to the dark text, right, are going to be releasing more neurotransmitter. And any of your photoreceptors that are primarily seeing the white background, the white screen, are releasing less neurotransmitters. So the photoreceptors that are detecting the dot think it's dark and release more neurotransmitter, and the photoreceptor that detects the right screen say it's light and releases less neurotransmitter. So in this way, the different photoreceptors in your eye, some of them are seeing, like you're looking at this slide right now, this video right now, some of them are seeing dark spots. They're releasing neurotransmitters, the dark text. Some of them are seeing the white background and they're releasing less neurotransmitters. And so that means the next neurons in your retina and eventually in your brain can tell the difference. They can tell the difference between the dark text, where the neurons are more active, they're releasing more neurotransmitters, and the white background when it's light, and so those photoreceptors are releasing less neurotransmitters. And so in that way, they can interpret this information and know, hey, over here there are dark shapes, and over there are just the white background. I do want to note, so we've talked about plasticity. I do want to note that there's also plasticity in phototransduction. Photoreceptors can adjust their average activity levels to match the level of light in an environment. You've probably had the experience where it's bright outside and then you go inside into a dark room, and at first you can't see anything, but if you wait a little bit, your eyes will adjust to that level of darkness. So that is called light adaptation. How does that happen? That happens primarily with the calcium coming in through those cation channels. So light levels over time will change the calcium levels, and the calcium levels will adjust the amount of actually they adjust a number of things, such as the amount of cyclic GMP, how well opsin activates transducin, and the conductance of the cation channel. I'm not going to explore this more. I just want to tell you that this is a thing that you should know about. All right, so that is phototransduction.